All right, welcome back everyone. Here we are starting block 2. We're past endocrine, but please don't flush that information from your head because it will come back several times throughout the semester. Now, as we move into blood, there is something that I always like to point out in that blood seems like it's a pretty small topic for an anatomy and physiology course, but in reality, blood is kind of the central underlying theme of this entire class, and everything that we talk about in Bio 139 is going to relate right back to blood. When we were in endocrine to start with, well, how did those hormones get around the body? In the blood. In the next block, we're going to talk about the cardiovascular system, and that's how we move the blood around the body. Later in the semester, we're going to talk about the respiratory system, and that's going to play a role in how do we oxygenate and deoxygenate the blood to get those gases to and from the body tissues. We're going to talk about the digestive system and how we move nutrients into the blood. We're going to talk about the urinary system and how we cleanse the blood. We're going to be talking about blood in some way in every single chapter. So, as we go through this chapter, don't brush it aside as something that seems like it might not be super important, because in reality, it actually is extremely important. So, let's go ahead and get started as always with our not really attendance questions. First, what class of tissue does blood belong to? And second, what are the two types of pressures involved in osmosis? So go ahead and answer those questions. Hopefully you've done that, and let's see what the correct answers are. What class of tissue does blood belong to? Think back to the chapter 4 in Bio 137, or that big tissue lab. Blood is a connective tissue. And what are the two types of pressures involved in osmosis? There was hydrostatic pressure, the pressure of water or other liquid against the walls of its container, and osmotic pressure, the pressure of the solutes pulling water back towards themselves. Remember, solutes suck. So these two are both going to play a role in this chapter. All right, so the functions of blood. Blood has a lot of functions, primarily transport, regulation, and protection. Let's talk first about transport. When things in one part of our body need to get to another part of our body, chances are they're going to travel through the blood. Blood transports stuff to the tissues or from the tissues to different parts of the body, from different parts of the body. So let's look at the two. Blood carries nutrients and oxygen to the tissues. It also carries those hormones from the endocrine to various parts of the body. Blood also carries waste away from the tissues. It carries carbon dioxide, which is just another waste, away from the tissues. Blood plays a lot of regulatory roles. It regulates our body's temperature. It does that by when we're cold and we shiver, it carries that heat that's generated by the muscles to our body to warm us up. It also, the small capillaries in our skin constrict so that not as much blood gets to our skin. That way we're not cooling that blood down when we're out in the cold. But if we are hot and we start to sweat, those same capillaries dilate to let more blood towards our skin surface. And as we sweat and it cools our skin, that then cools our blood, which then circulates through our body, cooling us down. Blood regulates our pH. So we have a very specific range of pH, and if it gets too acidic or too alkaline, blood carries a buffer called bicarb or bicarbonate that acts to maintain our pH of our blood. Later in the semester, we're going to see how we breathe to regulate the amount of CO2 in our blood, which in turn regulates the pH of our blood. And fluid balance, that osmotic pressure that we just talked about. The pressure of the liquid in our blood pushing against the blood vessel walls. Well, we would just call this blood pressure, but that is osmotic pressure, and that liquid in our blood is pushed out into our tissues. Blood plays a protective role. So, for example, hemostasis. Now, this is not homeostasis. This is a different word. Hemostasis is blood clotting. Blood clotting. And that sounds like it's a bad thing, but it's not. If we got a cut and we didn't clot, well, even a small cut, we would bleed to death. And we are actually getting very small cuts in the smallest blood vessels in our body constantly. That just happens from movement. And if it wasn't for hemostasis, we would bleed internally and not even realize it. It protects us from infection. We're going to talk about white blood cells later in this same chapter, and the white blood cells are a big part of our immune system, and they fight off infection. So let's talk about just the composition of blood. This is going to be a very short lecture because we're just doing a blood overview. Later lectures in this chapter are going to focus on the specific parts of blood. So what is the composition of blood? Well, blood is a connective tissue, like we just said. It is from the same origin as the other connective tissues, but if we look at the makeup, it's got the same structure as connective tissue. Here is a microscope slide of blood. Hopefully this looks familiar from Bio 137. Remember some of the features of connective tissues. They were not very cellular compared to some others. Remember muscular tissue and epithelial tissue. The cells were packed very, very tightly together, and there wasn't really a lot of extracellular matrix. Well, here we can see these pink donut Cheerio looking guys. Those are red blood cells. These guys that's got the big big purple blob in the middle of them, those are white blood cells. And then these little flecks that kind of look like dust on the lens, well, those are platelets. But we see a whole bunch of this white area here, and this is plasma. This is the liquid part of blood. And in any other connective tissue, we would call the plasma, we would call it the extracellular matrix. So blood is a connective tissue. But it's common to do something called fractionalization. And if you've ever donated blood, you might have seen this happening. If you've ever worked in a phlebotomy area, you've seen this happening. And what they will do is they will draw some blood and put it in a small test tube. And then they will put the test tube in a centrifuge. And a centrifuge is something that just spins around really, really quickly. And whatever's in these test tubes, it's going to separate the components out by density. The denser things are pulled towards the bottom, and the things that are not quite as dense remain near the top. And when you do that to blood, you get three regions. You get this dark red area at the bottom, a kind of clearish area right in the middle, and then the top is kind of a clear yellow. It almost looks like urine. And later in the semester, we will see that it is very, very similar to urine. So here's what it actually looks like. Over on the left is the artist rendition like we just saw. Over on the right is an actual fractionalized blood sample. At the bottom, here's that dark red area. Now, this area is where the red blood cells are. The red blood cells make up the bottom portion. And the red blood cells, if we took a measurement, that measurement is called the hematocrit. So this red area down here, this is where all the red blood cells are. And the measurement of red blood cells in a sample is called the hematocrit. So later in this chapter, we will talk about hematocrit. That's what that word means. Here in the middle, we've got this kind of clearish area. It just kind of looks faint red here, but you can see it's a little bit clearer. This is called the Buffy coat. And the Buffy coat is where we find the white blood cells and the platelets. And then at the top, that clear yellow, this is the plasma, the liquid part of blood. And it's mostly water, of course, and it's got a lot of proteins and ions dissolved in it. So, here's back to our artist rendition. And in the blood sample, typically, if everything is going correctly, we should have a hematocrit. Remember, that's the amount of red blood cells, the hematocrit, of about 45%. If it's higher or lower, that could indicate there's something wrong. The plasma should be about 55%, and again, if it's higher or lower, that could indicate something's wrong. And the Buffy coat, the white blood cells and the platelets, should be less than 1%. Now, if any of these numbers are off, that could be something that we could use as a diagnostic to figure out if there's something wrong with the patient. So now, the composition of blood. Here, we see the normal values. Now, when we talk normal values, there's kind of a lot of leeway there. What's normal for one person may not be normal for another person. What's normal for one group of people may not be normal for another group of people. So when we come up with normal values, well, what we find is we have to figure out who are we using as the ones that we're going to give the values for. So when we hear these normal values, especially in our book, they are talking about normal values for the average, healthy, adult, white male. Now, what that means is if we have someone who is not average, who is either well above average or well below average, these numbers will be different. If they are not healthy, if they have some sort of medical issue, well, these numbers will be different. If they're not an adult, if they're an infant or a young child or an adolescent or elderly, those numbers may be different. If they are not Caucasian, if they're not a white person, these numbers may be different. And if they're not male biologically, then these numbers may be different. So, the normal values for blood, when we look at it, it should be bright red if it's taken from an area that is oxygenated. It should be dark red or almost maroon if it's from an area with low oxygen. So most arteries are oxygenated, so most arteries will give us bright red blood. Most veins are deoxygenated, so veins will tend to give us dark red blood. We will learn in cardiovascular that there are some arteries and some veins that don't fit that. The average healthy adult white male should have about 5 to 6 liters of blood in their body. And the pH of that blood should be between 7.35 and 7.45. So slightly alkaline. Remember, 7 was pH neutral. Below 7 was acidic, above 7 was alkaline. So 7.35 to 7.45 is slightly alkaline. Now, if the pH goes below 7.35, we say that that is acidosis. If the pH goes above 7.45, we say that is alkalosis. But pH should be between 7.35 and 7.45. Blood should make up about 8% of body weight. Now, that will bring us to the end of this lecture, and like I said, this was a very short lecture. This was just to introduce us to blood, but we will be going into plasma next, and then after that we will spend a little bit of time with the red blood cells and the white blood cells and the platelets. We will look at some of the functions of blood and the makeup of each of those components. So, if you have any questions, let me know. Otherwise, I will talk to you next time.